now we're starting. Last lecture, we'll be talking today about information content. We'll be talking about errors in biology. And we'll start directly with a question. So if I'm comparing genomes in terms of the information, I'm interested in comparing Homo sapiens to E. coli. And so my question to you is, if I'm comparing in terms of genome size, or if I'm comparing in terms of gene number, what would be the ratios between these two very distinct genomes? So would it be about 5,000-fold, 500-fold, 50-fold, or 5-fold? Probably all the x's would be better suited here. And similarly, in terms of genome number, is it about gene number, about 5,000-fold, 500-fold, 50-fold, or 5-fold? Is the question clear? So we're comparing between two organisms that we study quite a lot, ourselves and our pets in our gut. And so what would be the ratio of the genome size and the ratio of the gene number? OK, ready to vote? So who's voting for A? The ratio of 5,000. OK, about half the class. Ratio of 500, the other half. 50-fold, maybe a few. 5-fold. OK, we're now moving to gene number. Who's voting for 5,000 more in the Homo sapiens? 500-fold, 50-fold, about a third, and 5-fold, about a half. OK, I'm happy that I asked. So let's talk about that briefly. Should we do it one way or another? OK, so let's start with genome size. What do we know about the genome size of the Homo sapiens? Anyone knows his genome? Right, so genome size of Homo sapiens, that's about 3 giga base pairs. And E. coli, our friend, how much? So it's about 4 point something. It's closer to 5 million. Somewhere between 4 and 5 million. OK, so if we're looking at the ratio of this, it's closest to 500-fold. And now we could also ask, not about the genome size, but about the number of genes. So how many genes do you carry? So that was one of the big puzzles that got an answer from the Human Genome Project. So before the Human Genome Project, if you looked at the estimates, they were all over the place. There was actually a contest of people trying to guess. I think it was called Gene Sweep or something like that. There was like an open website for people trying to guess who would be closest to the number of genes that would be recovered. And the values, they ranged by a lot. People were thinking above 100,000, smaller than the actual numbers, all sorts of things. In the end, it boils down to something on the order of 20,000. And the number, of course, is not still final. It's a question of how do you annotate, how do you define exactly. But the fact that this is the number was a big surprise at the time, mostly because of the contrast to what was already known. Anyone knows about E. coli? So that's somewhere around 4,000 to 500 genes. So this was already known roughly because, of course, the genome is 500 times smaller. It was easy to analyze it. There was lots of knowledge about it. And people were expecting that this number would scale in some proportionate way. But actually, it was a huge surprise to find that the answer to this is about 5-fold. OK, so this is starting about the information content of genomes. And now what explains this, this variation of about 100-fold? So I hear gene size, I hear introns, gene density. What? Operons, polycistronic operons. Anything else? Regulation, splicing. Something else that we have in the human genome that is retroviruses. Yeah, so all sorts of transposable elements. OK, so the two main things that I would mention in terms of trying to reconcile this 100-fold difference, the first one is actually transposable elements that take over more than half of the human genome. OK, so a very, very large fraction is built of all sorts of different, either ones that are still transposing or ones that got transposed and got stuck there. But the large fraction of the human genome is taken by those elements which are not genes per se. The other thing that we already discussed in previous lectures is that if you look at a specific gene, in an E. coli, you'll only have the gene itself. In eukaryotic organisms, you have it as exons interspersed with introns. And on the average in a human gene, the introns would be about 30 times longer, the introns, than the exons. OK, so if we're talking about a factor of 100, I would say, first of all, you have a factor of 2 that gets resolved by transposons, or even more than a factor of 2. And then there's a factor of 30 coming from the ratio of the introns to exons. And that sort of like helps you understand this large variability. OK, and of course, there's a lot more to be said. I would say in general about the talk that we're going to do today, as you can tell from the subject, it deals with issues that touch very closely on evolution. And in trying to deal with evolution, making generalizations is very, very difficult. So take that as a caveat to the things I'm telling you. I'm trying to give you the key numbers or the orders of magnitude of some of the phenomena. And there's all sorts of finer details, definitely when thinking about, for example, what happens with the human genome, which is extremely complicated and has all sorts of different components. So starting with that, I'd now like to suggest the following question. So in thinking about rule of thumb, about genome content and information density, I want to challenge you for the following. Let's think about the genome of an organism, of a bacteria called Vibrio cholera. So this is a bacteria that maybe you haven't worked with, but has had quite an impact on human history in all sorts of different ways. And we now know that the genome size is about 4 megabase pairs. Very, very close, like something like 4.03 megabase pair. And then my challenge for you is, what would you think about the number of genes it holds? So you might already remember we were discussing such things about how to think about genome size. But here I want to give you the following options. One would be any number smaller than 3,600, let's say. OK, so that's anywhere between, say, 1 gene to 3,600. B would be somewhere between 3,600 and 4,400. And C would be any number larger than 4,400. So as you can tell from the ranges that I'm dealing with, now it's a challenge not only regarding what kind of estimate would you make, but also how willing are you to bet in terms of the accuracy of your estimate. OK, is the question clear? OK, so who's voting for A, smaller than 3,600? A few. Who's voting for 36 to 4400? A few. Who's voting for more than 4400? Half the class. Okay, so this for me was an interesting question because I had this sort of like rule of thumb of about 1 KB per gene, but then how much are you willing to bet your money on how accurate would that be, right? That's the question here. Okay, so let's look at at some real data. So yeah, the quality is not that great here, but I want to do that quickly. What you can see here is a table. I'll try to read it to you because it's not very clear. It's just a compendium from all sorts of organisms moving from HIV, which has about 9 genes. And if you take its genome size, which is like 10 kilobase pair, and you divide by 1,000, you get a prediction of 10 genes. Okay, this is what would be the prediction if you were just taking the genome size, dividing it by 1,000, removing through 3 zeros. Then you could move to bacteriophage lambda, about 60 genes, and 50 would be the prediction. Moving to Buchnera, you'll have 610 and 640. Bacillus subtilis, 4,233 versus 4,200. E. coli that we're just discussing, is like 4,200. And from the genome size, it would be 4,600. And our cholera, which would predicted at about 4,000 genes, actually has, according to the current estimate, 3,949. So way better than 10% in terms of accuracy. This is all when looking at bacteria. When you start moving to Cerevisiae, you would have predicted 12,000, but you actually have closer to 6,000. And then when you go to Homo sapiens, you would have predicted 3 million, and you'll get only 19,000. And for wheat, you'll be predicting 20 million. Okay? According to these predictions, by the way, okay, before we move on to that, you could look at how it looks really for bacteria in different organisms. So here you see on the x-axis the genome size in megabase pairs and the number of genes as is currently known. And you can see how nice the data is, both for bacteria and for archaea. You can see how strong the correlation is. And the slope here is exactly of the rule of thumb, about 1 gene per 1,000 base pairs. And it's way better for most bacteria than 10% in terms of trying to make the prediction. So the eukaryotes are not on this figure. In terms of genome size, they would start somewhere here and go. And then where would the points be? Would they be above or below the diagonal? They'll be way below, right? Because as you increase the genome size from megabase pairs to gigabase pairs, you'll find that the number of genes increases much more slowly, and therefore they're well below. And actually, it sort of like seems like it's almost saturates at this 20,000, 40,000, whatever. It depends. Depends also on the fluidity of the organism. But what you would usually find is that it doesn't go, definitely not as fast as the genome size. In terms of looking, by the way, the genome size, anyone knows about the largest genome? So we looked at wheat as an example. So wheat is about, if I'm not mistaken, something like 20 gigabase pairs. It's a hexaploid. So it's not 2N, but 6N. It's a very interesting genome. But you can go further than that in terms of genome size. Anyone has other guess? Strawberry. I don't know what's the size of the strawberry genome. Do you know? Octaploid. Okay. So actually, the largest genome I could find information on is an amoeba by the name of Polychaos dubium, with the impressive genome of 670 giga base pairs. Okay, so it's about 200 times the size of our genome. I don't know. There was another question here?
I just wanted to ask, we're only talking about coding genes, right? No.
OK, so we're talking about genome size. You mean in terms of the number of genes? Yeah, we're talking about protein coding genes. Thanks for clarifying that, yes. Yeah, so in terms of how we're defining here, protein coding genes, yes. Good. So from this, I'd like to sort of like move into talking about this. All these genes are information that is coded on our genome. And I'd like to have some survey on what it means in terms of the accuracy of this information as it's being coded on the genome and how it's being used later on. So in thinking about different processes for which we might have errors in biology, what are some of the processes you would consider that could have errors in them? Yeah, so I have genome replication. Then I heard transcription. Then we have translation. Well, and then we have a mess of other processes, post-translational, that could have all sorts of different other issues with them. And so let's try and move between these processes and talk about something about the error rates in them. So we'll start with replication. Well, actually, I'll start with translation first. So translation is on the order of 10 to the minus 3 to 10 to the minus 4. And the relevant units here are, so it's like number, when I'm talking here, I'm talking about errors per codon. Meaning in the translation machinery, in the ribosome, in terms of incorporating the amino acids, you'll have on the order of 1 in 1,000 to 1 in 10,000 mistakes. Instead of putting the correct amino acid, putting an alternative amino acid. In transcription, it would be on the order of 10 to the minus 4 to 10 to the minus 5. So this is errors per nucleotide. And then in replication, it would be on the order of 10 to the minus 9 to 10 to the minus 10 errors per base or nucleotide. And this is, if you wish, this is for one. So this is for one act of translation, one act of transcription, and this is one act of replication. And this value, as you can see, is way, way lower. This is achieved by all sorts of interesting processes. For example, one important process, anyone knows, that enables one to achieve such low error rates? Exactly. So this thing called proofreading or kinetic proofreading. Oops. No. And then in some organisms, you wouldn't have this kind of proofreading. For example, in viruses. So in viruses, for example, which does not have necessarily such processes, the error rate would be significantly higher, on the order of 10 to the minus 4 to 10 to the minus 5 errors per nucleotide. And that seems to act in their favor in many times in terms of their ability to mutate pretty quickly in order to deal with all sorts of mechanisms of the host and to evade all sorts of processes used by the host. Yes?
Do you take into account the DNA repair mechanisms?
So the question was, does that take into account the DNA repair mechanisms? And I think it does, but actually one should verify. I'm not absolutely sure. I think it does. OK, so in thinking about those numbers, we usually want to have some intuition. So let's try and develop our intuition on that. In hand with discussions that Niv started in the course about trying to get intuition on information. So what would be a good way to think about errors? What other mode of saving information do we have? So for example, we're having books, right, or pages of paper just like this. So let's think about what it means to have, say, something on the order of 10 to the minus 4 mistakes. What does that mean in terms of having errors, let's say, in writing something? So if I have a page of paper, let's say it has 50 characters per line, and let's say I have 50 lines per page, it means that I have on the order of, say, 2,500 characters per page. So if I have 10 to the minus 4 errors, that means that I'll have 1 error for every 4 pages, right? Mistake somewhere in every 4 pages. On the other hand, if I'm talking about, say, 10 to the minus 10, that would mean what? That would mean if I have it every, if that was 1,000, that would have been 10 to the minus 7. So I could write something like 4 million pages without making on the average just 1 mistake. Okay, that means like it's like, I don't know, like a volume of Encyclopedia Britannica, 1,000 such volumes next to each other, and then in all of them there's only 1 typo somewhere within that. Okay, quite impressive in terms of what these molecular machines are able to do. And remember, they do it also, they have to make that, to replicate that information pretty quickly. Anyone remembers roughly at what rate? Right, 1,000 per second. So you have to write the Encyclopedia Britannica, 1,000 volumes of it, without making even 1 typo, making 1 typo in it, and you have to do it at a typing rate of 1,000 characters per second. Okay, that's quite a feat.
If you take translation and you divide it for protein, then it doesn't look so.
Okay, so what you're suggesting is, let's see what happens when one wants to produce a protein. Okay?
What is the amount of protein which have mutations? Yes.
Okay, so let's try and estimate that. Okay, so the question that was raised here, given that error rate, say in translation or in transcription, what would be the expected number or chance of having a gene that would be that would have errors in it, right? Okay, so let's call a chance for a mistake here P, and then ask ourselves, what is the probability, what is the chance of having a mistake when you have n events that have to take place. For example, when you want to translate a gene. Okay, so what would be the equation for that? So if what I want to do is I want to check what is my chance of having, say, I'm doing translation, that in the end I would have an error somewhere in the translation. Well, an easy way to do it is to think that's 1 minus the chance that I would have an error, right? Okay, what is the chance for that? I can look at 1 minus p. So p is my mistake of a chance. 1 minus p, that's the probability that I would do the right thing, right? That I wouldn't have an error. Now, if I'm translating, say, a protein with 300 amino acids, what are my chances? This is the chance that I won't be doing an error in the first event. Then there's the same chance in the second event. The chance that I won't be doing any mistake in, say, 300 times is 1 minus p to the n. Let's keep the same n. So this is the chance that I won't be doing any mistake. And therefore, 1 minus then is the chance of an error somewhere in n events. Okay, you're with me about the, okay, so this is the equation for dealing with your problem. Okay, now let's try and plug numbers. So what would be numbers of interest to us? So let's call it E, big E for the error rate. So we might be interested in this for 10 to the minus 4 and 300 amino acids. So anyone could plug that, but this comes to about 3%. So this is the chance if you have this error of mistaken translation and you have 300 amino acids, 3% of them would have an error somewhere within them, and the rest would be fine. You can analyze the same thing if the error rate is higher, say 10 to the minus 3 for 300. And now you'll have about 26% with an error in them somewhere. Or you could think what would happen if you're looking not at translation, but at transcription with 10 to the minus 4 error, but now you need 1,000 bases. This would be about 9%. So first of all, I would say not all of those errors would necessarily make the protein non-functional, right? Some of them might be synonymous. Some of them might be ones that change the amino acids or change the nucleotide, but might not be something that would cause the protein not to be functional. At the same time, I think this also shows you something. First of all, you're getting a feel to how much errors there are, but actually there are quite a few quality checks mechanisms that are now being observed in recent years more and more, that, for example, many of the proteins do not finish translation. They're being detected along the way as ones that should be stopped translation in the middle and taken back to degradation. And that might relate to what you're getting here in terms of this calculation. Yes?
Maybe I'm just confused, but I think that this error rate does not take into consideration the complication of sometimes an error in a codon is not an error in the protein. Like if it's in the wobble position or
Yes. Okay.
the actual errors are much less.
Okay. So if I understood you correctly, you're saying that, say, in transcription, you might have some errors that actually do not cause harm in the sense that they're synonymous. They're just in the wobble location or something like that, and therefore in the protein would not change the amino acid. You're definitely right. Yes, I agree. That's true. Yes?
How can you characterize the errors? Are they uniform, or have they like a tendency to go together? In all of those.
Okay, so the question is about how much is this uniform or how much variability there is and whether things go together. So my guess is that probably nothing happens uniformly. There's all sorts of bunching and tendencies and correlations. This goes back to what I was mentioning, that I'm trying to make some generalizations. Also, those numbers, they're definitely not written, in stone. These numbers could vary depending on, say, whether you're in stressful conditions or in non-stressful conditions, in one organism or another. These are just rough numbers that enable us to make some deductions about what are we expecting in terms of fidelity versus errors. But I would definitely expect that there will be all sorts of tendencies here for things to, if you have errors, to have more errors adjacent to that and things like that.
Yeah, because in replication it has lots of implications. Like if it's like one gene that is mutated, or just like many genes that are mutated in many areas.
Right, so each such thing, yes, so that would definitely have implications. By the way, you can compare that to what is being achieved today so we were talking about books. So making, 10 to the minus 10 errors in books, writing 1,000 volume encyclopedia and making only one typo, that would be very, very tough. But in terms of data storage, today, what Uri taught me was that you could get 10 to the minus 14 as the benchmark of what is being asked for in terms of data storage. That means that you have only one mistake in like a few hundred tera bases or tera bits of information. So in terms of that, we've been making quite a lot of progress. So let's take these numbers and see how they could help us in trying to understand what happens in cases of interest that we study in the lab. So think about an experiment that maybe you're often doing by just putting some 5 ml overnight culture of bacteria. And let's say you're interested in the following. For some reason or another, you need to have, in the morning, after you put this for you started the night, with just throwing in one bacteria inside your test tube, and you let it grow overnight. And even though you started with something that came directly from ATCC, and the genome is, exactly like in the textbook or like on the website, you really need to have some single base pair mutation happening at least in one cell, which is what you're going to test. Let's say you need an A to a G in, I don't know, location 2,507,423. You want to have this exact substitution. And the question is, what do you need to do after you finish with the course? And let's give you 3 options. One is that you have to do random mutagenesis, meaning that you'll have to expose them to, say, UV radiation or EMS or anyway, your favorite random mutagenesis. The second option is that random mutagenesis just wouldn't cut it, and you need to do site-directed mutagenesis. Or you can really go directly to this website, to this location. And the third option, it is already there in the morning. No need to do anything. Yes?
How do you plan to isolate this mutation? Or
OK, so I didn't think far enough. I don't even know how I'm going to isolate the mutation. Probably there's some way to put it on some Petri dish, and only the cells that have this base pair would be able to survive.
So you just need one cell with this.
Exactly. But it has to be at a very specific location with some specific mutation. So who's voting for A, for doing random mutagenesis? Who's voting from site-directed mutagenesis? Who's voting for the mutation already being there? OK, so beyond your intuition, which I think will get you pretty far, let's try and put some numbers behind it and try to analyze what would happen in this dynamics. So we have a bacteria with the genome, which is replicating all the time. And so what do we know about the genome length? Well, we've just been through that, right? We said about 5 million bases. So this is on the order of, let's say, 10 to the 7 bases. OK, it depends whether you're calculating here in terms of bases or base pairs, but I don't think you should worry about a factor of 2 here or there, as you'll see also in a minute. OK, now we have the mutation rate. Let's call it mu here. Let's say it's on the order of 10 to the minus 9 error per base per replication. So now if I want to know what's the chance when I have an E. coli that's replicating that has a daughter cell, what are the chances that a daughter cell would have a mutation somewhere in its genome? Right, so it's 10 to the minus 2 to the 7 times 10 to the minus 9. So on the order of 10 to the minus 2 mutations per genome, per replication. So that means that not only would, in the daughter cell, I probably wouldn't have that, that mutation that I'm interested in, but actually throughout the genome, it's almost what's known as fidelity. The accuracy is such that you have only one percent chance that a cell would have a mistake somewhere in its genome. So high is the fidelity of the DNA replication process. Okay? But what plays in our favor in terms of achieving mutations? It's the fact that I have lots of cells, right? So we said we have here 5 ml. And you know, after an overnight, it gets into saturation. So there's quite a lot of cells here. And you know what? Let's even not think. OK, so how many cells are there in this? So let's say OD here. What? So the OD in saturation could be, I don't know, 5, 10, 2. Let's even be, let's say the OD. We said we have a volume of about 5 ml. Let's make an OD conservative one of 2. OD 600. So how can we move from that to knowing something about the number of cells? Does that ring a bell from some time? Right. So, OK, so we had a rule of thumb that goes somewhere along the path, along the lines of about 10 to the 9 cells per 1 ml at 1 OD. And as we said, that could vary. Sometimes it might be closer to 10 to the 8. Sometimes it's more than 10 to the 9. But that's a characteristic number. And as you might remember, we were comparing that to what would be the fraction of the volume that they would take over. That's about 1 in 1,000. And we're comparing that to the density of the students in the class and all that. But let's see how this, from a few lectures back, helps us into seeing what would happen here. So if I had 1 ml at 1 OD, this would be 10 to the 9 cell. But I have 5 ml with 2 OD. So how many cells do I have roughly? So I have about 5 times 2 times 10 to the 9. So that's roughly 10 to the 10 cells in my saturated culture. Now, we started with 1, we went to 2, 4, 8, 16, 32, etc. But we could think even just in what happened just in the last replication, right? Most of the mutations happen when there's a huge population size. So in the last step, you went from, say, 5, 10 to the 9 to 10 to the 10. So you had on the order of 10 to the 10 replications just in the last round, or a factor of 2 less than that. And we said that in each replication, we have on the order of 10 to the minus 2 errors per replication. Therefore, we'll have about 10 to the 8 errors that have been produced in the last recent replication in the culture. What does that mean? We know that genome is on the order of like 10 to the 7. It means that each and every location on the genome has been hit several times, say, I don't know, like 10 times, with new, what's known as Denovo errors that have occurred just in the last previous stage of replication. Therefore, I would say that if you're interested in having this kind of mutation, you're probably fine by just expecting that it would already be there. Yes?
Couldn't you get that answer without knowing how many bases there are? Is it like the bases cancels out?
Lovely. OK, so you're suggesting an alternative way of doing it. You're saying, why do you have to go through the number of bases? You already know that you have 10 to the minus 10, or what did we take that, or 10 to the minus 9? 10 to the minus 9 errors per site, right, per base. And if you also have 10 to the 10 cells, meaning this is 10 to the 10 replications, you can tell already from that that you'll have on the order of 10 new rising mutations. I agree completely. You can do it that way. It's more elegant. The reason I chose not to do that is because I wanted to go through the step that tells us something about the fidelity in terms of the accuracy of the overall whole genome replication, and to find this interesting contrast. On the other hand, at the stage of the cell replication, even though it's a long genome, your fidelity is extremely well. There's almost no mistake whatsoever. But because of the numbers, at the level of the population, you'll have an error. But I agree that this is a more elegant way of doing that. Yes?
Your error means that you changed your nucleotide to any other nucleotide. You want specifically to a G, so that will increase it.
Thanks for that. So I cheated you by a factor of about how much? About 3, because I was moving. I didn't say that I want to get a mutation here. I want to get a specific mutation to G. So there's another factor of 3 here. Thanks for reminding me that I should say that. So it's not a coverage of 10. It's on the order of coverage of 3 or whatever. Okay, what would happen if, I'm not interested in cells, but I'm interested in human beings, right? So let's think about the same question that we have here. But now I'm still interested in a single base pair mutation that's supposed to, save humanity from some, new Ebola or whatever, some big problem, and we need exactly this kind of mutation somewhere in the genome. But now, as you know, it might not be 2,500,000. It might be, because the genome is much larger, it might be in location like this, because we have, as we said, 3 billion base pairs where it could happen. And now the question is, if we're trying to look for that specific mutation, obviously we're not going to do UV or EMS on the human population, but would we expect to have that in the human population? Let's say in terms of things that happened in the recent, only new mutations that happened in the recent, in the Denovo, in the last generation. Well, that could be done in the elegant way that was just suggested here. We have on the order of a mistake of about, say, again, 10 to the -9, right? Then what is the population size of humanity? So about 7 billion people. That would bring us, that's probably 10 to the 10 from cells. This would take us to humans. Which means, again, that we'll have a coverage such that every de novo mutation that we might be interested in order to save humanity has happened de novo, just in the last generation, is being carried by somebody somewhere on Earth. Anyone sees something that I forgot? There's some mistake here along the calculation. Can you spot what it is? There's a missing factor.
Some minor tweak, but some of the errors would result in non-viable organisms.
Okay, so I didn't talk about it all until now about the fact that I'm thinking about say neutral mutations. Obviously, if there are mutations that are lethal, they won't be surviving either in the culture or as humans. That's definitely true. Anything beyond that in terms of thinking about what happens in terms of humans and the error rate?
Maybe it could be in the intron, so it won't be much effective.
Right, so it could be in the intron, but I'm interested now in a specific location. So this was the coverage for specific locations.
It would be one cell in the human.
Okay, so there's something with the cells in the human.
The fact that we're diploids?
Yeah, we're diploids. I'm not getting into that.
Genomes in a human. So this is in the per genome.
Okay, so it's per genome. It's fine because I'm interested in per genome, but
The number of cells may be important.
okay, so there's something with the number of cells, right? So what is with the number of cells? Everything that we analyzed in terms of the error rates was the error per nucleotide per replication, right? But how many replications would we have in a human being from the mother or from the father to the progeny? It's not one replication, right? Because you're multicellular and you need to look at the gametes, both at the sperm cell and the egg cell, see how many rounds of replication has happened until you reach the gametes, right? So there's a factor that has to be added here.
But it's only one for the one of them, so it doesn't really matter. It's this percentage.
So in terms of looking at how many, say per human, how many errors would you expect to have in a generation? Say 10 to the minus 9, that's per replication. But now you have to multiply by the number of replications. So where would you think the errors that you're beginning as a human would come from? Will it come from your father or from your mother? So it's not identical, right? Because you're getting either, the question is whether you'll be getting it from the line of the gamete that leads to sperm or the one that leads to eggs. So the values that relate to that, so we're interested in the number of replications per generation. And so for females, the characteristic number is about 20 to 30 replications per generation. That's from that's looking at the line of production of the eggs, of the ovum. And then you can compare it to what happens in males. So in males, in producing sperm, that really depends on the age. So that would go from roughly, say, 35, that's at age 15, to about 800 at age 50. So as a generalization, I would say the number that was given here for the number of errors has to be multiplied by another factor of about 100, which is the number of errors or number of replications per generation. So instead of 10 to the minus 9, you'll have about 10 to the minus 7 because there were 100 replications from your parents' gametes to your gametes. And then you can also see that, if you're interested in some specific mutation, you'll probably be getting it from your father, for better or worse, rather than from your mother. Yes?
What about the 10 to the 14 cells?
Right, exactly. So we also said
after discarding all the apoptosis.
Right, so what we're talking here is about, so what about these 10 to the 14 cells? There definitely we could look at the number of replications that would lead to them. So there we're talking about mutations that are known as somatic mutations, one that are happening, in some lineage of the cells in your body, what would not be transmitted in terms of evolution to the progeny. So that's a different question. That's also, it's the same numbers, and one could analyze how many replications are involved in that, but that would not govern what would happen in evolution further down the line, because that does not exist in all of your cells or in your gametes. By the way, if you're interested in looking how much, let's just do it very quickly, how many, on the average, a cell in our body, how many replications did it pass through if everything was happening, like just homogeneously as like a binary tree from 1 to 2 to 4 to 8. So the number currently estimated for the number of cells in the human body is about 3 times 10 to the 13 cells per person. Okay? Just to make it nice and round, let's go with your number, 10 to the 14. Okay? In order to know how many replications, I wouldn't want to have the base as 10. I prefer to have the base as 2. Right? So that's on the average, not very far from if I want to have instead of 10 a 2. So I'll take 3 point something or 3. So that's 3 times 14. So it's about 2 to the 50 or something like that. Okay, so on the average, if everything was going like a binary tree, you'll have 50 replications to give you a whole human being from the original merging of the sperm and the egg. Definitely that does not work exactly that way because we have stem cells and everything is way more complicated. But it's just a fun calculation. Yes?
What about the combinations?
Yeah. So
With the condition
Right. So I didn't talk here about all sorts of issues that relate to diploids and recombination. I'll mention them maybe briefly afterwards, but basically we're not getting into that. That's also a very interesting rate that should be added here in terms of recombination. There's also the rate of deletions and insertions and things like that. And there are also numbers on that that one could collect and analyze. Okay, so things seem everything is pretty available. Let's ask the following question though. Let's say I'm looking not for a single base pair mutation. Let's say I'm looking for a double base pair, Denovo mutation. I'm saying here Denovo because I'm not interested in, so I'm going back to the human population, and I'm interested now, it turns out that one single base pair substitution would not save humanity against Ebola, but you need to have two specific mutations. So the other one would be at, 1 billion somewhere here. And now you need to have both mutations in order to save us. And the question is, how long would that take? And I'm saying de novo because there's all sorts of fine details otherwise in terms of the existing variability that already exists in the human population, so I don't want to deal with that. I'm thinking, okay, starting from the reference human genome and looking at replication, say, of that in terms of the error rate, how long would it take until I would be getting a double base pair mutation? So my suggestion for you would be that some, say, newborn in China this year has it. Or alternatively, a person alive somewhere on Earth has it, this specific double mutation, or it will take millions. Millions of years. Question clear? Who's voting for the newborn in China that probably has it? A few people. Who thinks that the person alive somewhere on Earth has it? A third. And who thinks that it will take millions of years? A few others. Okay, so let's try and look at how we could approach that. So we already know that for every specific mutation that we're interested in, we have it in about, we said we're covering it in a replication for humanity in about 10 times. Or if we take into account the amount of replications, it's more than 10 times, right? Maybe 1,000 times. Okay? So now let's break down the problem into two sub-problems. The first sub-problem is getting the first mutation. Okay? So we said the first mutation, that would exist in about 1,000 people on Earth in the last generation. Let's say out of all of humanity. But now we need another mutation on top of that. So we could look only within that population of 1,000, and we're looking for a specific mutation. Now we know what is the chance for that. The chance for that is in every generation, again, it was 10 to the minus 9, even times the 100 replications per generation, it's 10 to the minus 7. So if I have 100 people or 1,000 people carrying it, but now each one has a chance of 10 to the minus 7 of having that specific alternative mutation, the chances are like 10 to the minus 5. In terms of years, this would take us millions of years. So there's something interesting happened here. Even though in terms of getting one mutation that was relatively straightforward, two mutations that would take millions of years for the whole population of humanity. That also holds, by the way, for the overnight bacteria that we started with, right? Because we said that for the bacteria, we found that we have a coverage of about, say, 1-fold or 10-fold in our overnight. So if you need one mutation in overnight, that's easy, that's trivial, you'll definitely be getting it. If you need 2 mutations, you'll probably be needing all the culture of bacteria throughout Earth to, all the different masses being produced for a long time in order to get 2 base pairs just in random. So this shows, I think, something about the combinatorial complexity and how things increase so quickly when you're trying to look at more and more mutations. What could solve such issues if you're interested? If the only thing that would save you is having a double mutation, what mechanisms could help with that?
Diversity.
So one thing is diversity. If there's already existing diversity, that definitely could help. Meaning, if, for example, you have such a diversity in the population that there's a good chance that, for example, one of the mutation is already existing as a neutral way in a large fraction of the population, and now you only need another mutation. Okay, so that's not so tough. Another option, not in Denovo, but if you have a set of mutations that is required, if each one has a fitness advantage, then we're not anymore in the domain of having everything neutral, but you're sort of like climbing a fitness landscape, such that if every mutation is beneficial, it would tend to grow in terms of its fraction in the population, such that now the chance of, on top of it, getting another one is now just like a single base pair substitution and not the combinatorial co-occurrence. What else could help you in achieving combinations of mutations? So sex, right? If you have sexual reproduction, that's one of the nice things. If you have sexual reproduction, you can collect mutations that happen in different lines. You don't have to have them all reoccurring in the same organism. They could be, this reshuffling enables you to collect mutations from the diversity that already exists in the population. Okay, so these are some of the examples of how, of the problem that one is dealing with, this large space of possibilities. Moving one step, that's relatively easy, you'll be getting in the lab anytime. Going several steps, that actually calls for all sorts of evolutionary processes that are non-trivial. We touched upon a few of them, but actually this, I would say, is still an open challenge of the different mechanisms that play in natural selection and evolution. There's all sorts of issues about selection for evolvability, what kind of mechanisms and situations would make it such that when you'll be dealing with a new challenge in evolution, that have a higher chance that the genomes that you bring into the game would be able to survive. As part of that, one could consider modulating these error rates, both at the level of replication, but also in other levels, under stressful conditions or non-stressful conditions. And this is all things that are so like at the cutting edge of what people are trying to analyze and understand in terms of how evolution is able to give us the diversity that we're seeing and dealing with changing environments. I think with this, at this point of making the connection between evolution and the numbers, that's a good point to finish the course. So I hope that was useful to you. We were trying to move from starting from looking at just the sizes, volumes, all sorts of things like that regarding cells, to talking about the concentrations prevailing inside cells, looking both at concentrations as well as the absolute numbers associated with them. From there, we were looking at energies and forces as another significant players inside cells that enable us to move into durations and timescales taking for processes inside cells. And we ended today with looking at the information in biology, the information content, and the error that happens along the process that also are not only problematic errors, but also beneficial errors in leading to diversity. I would say that there's several thank yous that should be said here. One is to Uri Bernholz, that was the teaching fellow of this course. Many of the things that you saw me do elegantly, that's because he came up with a suggestion before that and enabled me to say it in front of you. Another thank you is to a longtime collaborator of mine, Rob Phillips from Caltech, who's not here with us, but he's part of developing some of those ideas in the book that we're writing together on cell biology by the numbers. And also seeing him teach at Caltech inspired me to try and also try to teach as well. So I'm thanking him as well. Everything that you've seen here, and those that you haven't seen here, you can also watch from home, and that thanks to Itzik. So thank you, Itzik. And then I would say that the reason that I was able to explain things sometimes clearly is because for every lecture, I first had guinea pigs in my lab as part of the group meeting of trying all those lectures and getting comments from them. So I thank the lab members for giving good comments on that. And finally, I think from the feedback that I've been getting from questions from you, from your voting and active participation throughout the course, I think that was essential for the success of the course. So thank you. And I would not, okay, you definitely could clap yourself. And I guess my thank you would not come only as just by saying that, but also I would say that anyone who's interested to dig more deeply into that, I'll be happy to hear from you and seeing the assignments that you'll be starting to present next week. And as showing my thankfulness, I would also say after giving your final assignment, anyone who's interested in getting the book of the course that contains much more information than what we just did, I'll be happy to send you a PDF version of the book. So after you're done with all of your things, just let me know if you're interested, and I'll be happy to send you a PDF of the book. Okay? Thank you very much.